I'm going to discuss with you in the next 25 minutes or so our efforts towards monitoring the brain. I'm a neurosurgeon, and it is of obvious interest to me and my colleagues to have better ways to figure out what's going on in the human brain. When I was in my previous life in Cincinnati, we started a process of looking into this. And the way it started was, one day I said, I wonder what's going on in the Department of Electrical Engineering and Computer Science. And I met with one of the professors there just as an exploratory kind of meeting. And I said, do you have anything that would be helpful in monitoring the brain? And he said, well, I don't know, but I have this young postdoc who's actually a PhD student, Chunyan Lee, who's been working on some device. We are not quite sure what to do with it. And I met with Chunyan, and we started talking, and I took her to the ICU, and I showed her what the problems were. And really, what follows and what I'm going to present to you is really Chunyan's work, and she's sitting back there, so any and all questions will be directed to her. So with that background, let me just give you a little bit of context here. Brain injury, like if you're driving home this evening, you're in a car accident, your head hits the inside of your car on the steering wheel or any other part of the car, your brain is slammed into the skull like a piece of jello, and you have an injury. So that is the basic injury. But then in the next few minutes and the next hour or 2 days or 3 days, a whole bunch of stuff happens, which was not initially there, but what we call secondary injury. That leads to brain swelling. It leads to a whole bunch of biochemical stuff, which we only kind of understand. So we have the primary brain injury with the macroscopic, that is the big damage, and then all the secondary stuff, which is the microscopic injury and all the biochemical processes that occur thereafter. So one way to look at it is shown in this cartoon, where with the initial injury, you have the excitotoxicity. You're all familiar with NMDA activation, which allows calcium to get into the cells, and calcium's inside the neurons is not good, and it tends to kill the neurons, so you don't want the calcium influx. Then following this, you have the inflammatory process, and the first two talks you've heard a lot of discussion about inflammation and the role that it may have in a bunch of processes, including this. And all of this leads to neuronal death. And so the general idea is that if we are able to do something here that would reduce the amount of calcium influx, that we may be ultimately able to reduce both the inflammation as well as ultimately reduce the number of cells that die. So the general idea is you have a certain number of cells that are killed at the moment of impact, and there's not much you can do about that except, of course, airbags and seat belts and so on. But then, as scientists and as clinicians, we want to try to prevent all the other crap that happens after that. Okay, that's the secondary injury. But how do you know what is happening? We don't. We don't know what the hell is happening, and certainly do not know clinically from one point to the other, from 1 minute to the other. We don't know really exactly what is going on. So we kind of shoot from the hip and try to look smart. And the general idea is that we are hoping to be able to find a way to monitor this thing better so that we know what we are treating and we can treat specifically for whatever is going on with the patient. So some fundamental challenges. To date, although my whole career I've been interested in finding the single wonder drug, the silver bullet that will cure head injury or improve outcomes from head injury, the fact is that we have not really shown efficacy with a single drug so far. There's a progesterone trial that is completed, and the results are due in the next month or so. And we are hoping that finally we may hit a home run, but we haven't done that yet. A variety of drugs have been tested but have failed, and our ability to continuously monitor these physiological and biochemical changes has up until now been limited. So what do we use to monitor patients? We use a clinical exam or radiographic techniques. In other words, we examine the patient, we get a CAT scan, and in most good ICUs, we tend to repeat the CAT scan perhaps every day, sometimes more often, sometimes less. But so we try to look at what's going on macroscopically. But of course, this provides us a snapshot in time. We have certain bedside monitors which may be invasive or non-invasive. Most non-invasive stuff is basically bogus. It doesn't give you much. It can be continuous or non-continuous. So non-continuous is, of course, not as useful because these are snapshots again. And here's just a list of the different things that have been used. Just to kind of move it along, really the list of what we practically use is intracranial pressure, brain oxygen saturation. So intracranial pressure monitoring has been around for about 40 years or so, used sort of fairly regularly in patients with severe head injury, that is those who are comatose, but not in people with concussions or milder injuries. Brain oxygen saturation became available maybe about 15 years ago. It's used sort of in a patchy way. Not everybody uses it. Brain temperature usually goes with the oxygen saturation. Cerebral blood flow is, global cerebral blood flow is hard to measure, and hardly anybody does it. Neurochemistry, there haven't been a way to monitor that except for microdialysis. And that, again, is a very labor-intensive technology which perhaps in the whole country, maybe half a dozen places use it, primarily for research purposes. And finally, you have electrical activity, which typically has been EEG, and you get that when you're worried that the patient may be having a seizure, which incidentally, we find that a significant proportion of these patients do have seizures that may or may not be detected because often these patients are heavily sedated in the ICUs. So the limitations of these current monitors are you need multiple sensors if you're going to try to get this information. You may have to put in multiple insertions. In other words, you have to make holes in the head and put in a whole bunch of different probes. And obviously, with each probe you put in, there is some small but cumulative or additional risk involved in both hemorrhage as well as perhaps infections. So where we are going with this, as I mentioned, here's ICP and then the Lycox monitor, which does the brain oxygen saturation, and EEG, microdialysis, blood flow, as I mentioned, is not very commonly used. And what you end up with as you try to do this is it becomes more and more complicated in a situation where the patient already has all kinds of lines and stuff coming out of them. You now have even more, and it just makes it logistically and practically a complicated proposition. So the next generation monitor that we have been trying to develop for the last 6 or 7 years is as a single probe, we can monitor multiple physiologic, metabolic, electrophysiological parameters continuously and in real time. So the design that we have been working on is taking this, and I'll give you more details on this in a minute, but taking a flat strip which has a bunch of monitors in it, and we'll talk about this in a second, and make it into a spiral so it becomes kind of a tube, and I'll explain why that is advantageous. But the interesting thing about it is then you can monitor the fluid, the spinal fluid that is egressing out of the tube, You can monitor that, and you can monitor the outside of the tube, which is in contact with the brain. So you can see what's going on in the CSF, and you can see what's going on in the brain tissue. You can also drain the spinal fluid. One of the techniques we use is simply draining the spinal fluid to reduce the pressure inside the head. Because one of the bad things that happens with all the inflammation and the increase in cerebral blood flow as a reaction to the injury is the increase in intracranial pressure. And one way to reduce the intracranial pressure is simply draining the fluid out of the ventricles. So this allows you to do that. And if we were to accomplish this, it would be with one hole, perhaps less expensive. We can monitor both the focal, the tissue stuff, and the CSF. We can also deliver drugs through this if a drug became available that one might want to inject directly into the brain. And then one other major problem with the monitors has been that if you put in a monitor which has no fluid coupling, once you put that thing in, it's a leap of faith. You do not really know whether it is accurate or whether there's been a drift. Whereas with this technology, we should be able to recalibrate it any number of times so that we can be sure that when you're reading a pressure of 20, which is considered the upper limit of normal inside the head, 20 millimeters of mercury, we are in fact at 20 and it's not actually 40 and you're getting a false sense of security. So what we started to do was to use MEMS technology, which is microelectromechanical systems, a term which only an engineer could have made up, and combine that with flexible polymers to create what we call the smart catheter. The advantage of the polymer is that it is biocompatible and compatible with the MEMS processes, and it is something that the engineering people have developed a comfort level with. So, as I mentioned, you take a strip and then you implant a bunch of sensors into the strip, then you spirally roll them, and as I mentioned, you can have these sensors both on the outside as well as on the inside of the strip. This is in contradistinction to the traditional approach where if you could have a sensor on a catheter and then you'd have to have a bunch of wires coming from that sensor and going to the monitor outside. And those wires, of course, traditionally are somewhat bulky, and so the lumen of the catheter tends to get smaller, and it makes the device bigger and bigger, and you don't want that device to be too big because remember you're sticking it through the brain into the ventricle. So not necessarily smaller the better, but to a certain extent smaller the better. So the first generation of this design required a placement of these different sensors, and we thought that some of the things that we wanted to monitor included brain pressure, oxygen, lactate, etc. And the interesting thing here is you have these different layers which you can put the different sensors in different very microscopic layers so that you get separation of the sensors to reduce what is called crosstalk between the sensors. So you have these different sensors and you have these different holes so as to allow the sensors to be in communication with either the CSF or the brain, but at the same time be in a different layer of this device so that one sensor does not affect or interfere with the other sensors. Nevertheless, with all of this, we have a relatively small catheter size, and by doing the spiral design, you can get the sensors to be on a smaller area, that is 1 centimeter, for example, which allows you to put the catheter into the brain or into the ventricle, and particularly if you're devising the catheter as a laboratory experimental device, obviously a mouse or rat brain is much smaller than the human brain, and so you want to concentrate all your sensors in a very small part of the device. So this is definitely Chunyan Lee's slide, where you've got all of these different types of issues relating to these sensors. So you can have electromagnetic or coupling or chemical crosstalk, etc. These are all the challenges. I must tell you, when I first started working with Chunyan, we're going back, about 6 or 7 years now, I said, Chunyan, how long will it take for you to develop this device that you've been working on to be able to use it clinically? And she said 3 months. 3 months. I said, okay, what do I know? I'm a clinician. I said, 3 months sounds good. Now here we are 7 years later, still thrashing it out. But you can understand what the challenges are in making this all happen. So the current technology, intracranial pressure, these are the things that we wanted to measure. And just a list of current technology. I'm not going to read through it in the interest of time, but just to give you an idea, and the working principles, and I'll go over that in a second. But we thought that the important things to measure are intracranial pressure, brain temperature, blood flow. We've already talked about this, and you know that these things are important. Oxygen tension. Things that we have not been able to previously measure on a regular basis are glucose and lactate. We all know that when the brain does not get enough glucose or enough oxygen, then it's not good. It goes into anaerobic metabolism, and you get production of lactate. So if you are able to monitor the lactate, if the lactate is going up, then presumably something bad is happening, and it gives you kind of a canary in the coal mine or early warning system that things are not quite right. And electrocorticography, that's the ECOG, is a way of keeping track of the overall electrical activity of the brain, which again can give you a sense of how things are going. You understand, when the technology has not been available, we really have not been able to correlate bad stuff happening with some of these things. So it opens up a whole new world. If we are able to do this, we'll be able to really track the brain in real time, and we'll have a much better understanding, I believe, than we currently do. So the pressure sensor works on a piezoresistor principle, shown here. So you have the active and passive piezoresistors, and it turns out that when you have sort of a membrane here, that the most change in resistance is at the edge of the membrane, which to me seems counterintuitive. You would have thought it will be like at the point where it moves the most, but apparently that's not the case. And so that is with the pressure sensor. For cerebral blood flow, we've used a thermal diffusion mechanism where you heat the coil in one place, and you see how long it takes for the temperature to drop on a second sensor. And the more rapidly the temperature drops, the more blood flow there is. It's sort of a washing out principle, as I understand it. So that's the technology used for a local blood flow. Remember, this will only look at the blood flow perhaps for a cubic centimeter around the catheter. This will not tell you what the blood flow is in the whole hemisphere or in a more global sense. The biosensors, looking at the different chemicals, are used. What is used is an enzyme-based electrochemical mechanism. And interestingly, one of the advantages of this technology is that it can function even in hypoxic environments, because some of the other technologies, if there's not enough oxygen, the system doesn't work. And then the electrocorticography looks at direct current voltage, and it uses a device which has layers of iridium, platinum, and gold. And you wonder why it costs so much. But the advantages of this are that the impedance is lowered 17-fold, the polarization rate is lowered 80 times, and there's lower low-frequency noise. So, so then in summary form, the different generations, we are still working on completing the first generation, which will have the ones which are listed here. And then as we go forward, we can add pyruvate, glutamate, glycerol, pH, biomarkers, and virtually anything that you think is important could potentially be added on. The only thing I would warn you is that, as Chunyan told me, it would take her 3 months to get the device. I think each thing that you add on is probably a couple of years of work. But nevertheless, it is doable, and it makes one excited to think of the different things that one could potentially add on. So here's what the Gizmo looks like at present. This is a small one for rat brain monitoring. And you can see that it looks like a tube. You can see all the different sensors which are in the wall of this tube. And that sensor is connected to this device, which is known as the head stage. And the head stage brings in the information from the device, is then connected to this signal enhancement device, which is shown here. That is connected to a signal interpretation device, which is here. And that is connected to the monitor, which you typically, when you go into an ICU, you see a monitor above the patient's bed. That would be over here. So it goes through a couple of different processing units before it can be connected to the monitor, which one would think of as a traditional ICU monitor. Now, this has been looked at using a rat middle cerebral artery model, which is in Dr. Ping Wang's lab, and it has shown that when you cause a stroke, there is about a 70% drop in cerebral blood flow with the smart catheter implanted in the hemisphere which is being stroked out. You can see all the changes that occur. So this is a screenshot of what one might see in a printout coming from such a device. You can see that when the hemisphere is caused to stroke, you will get a bunch of different things happening. You will get, for example, a drop in brain oxygen, a drop in brain glucose, a subsequent increase in lactate, and an increase in temperature, and an increase in intracranial pressure. On this side of the screen, you see the different forms of electrocorticography. And here you can see that associated with ischemia, there's a definite drop-off in brain electrical activity. All of this is shown more clearly in this slide, where, again, you see the EEG drops. This is where you get the ischemia. The DC potential, which is the low frequency waves, also known as spreading depolarization or spreading depression. You can see how that occurs. You have a big wave of spreading depolarization. You get a drop in oxygen, a drop in glucose, elevation in lactate, temperature goes up, blood flow goes down, ICP goes up. Now, this is very preliminary, but look at this. The ICP does not go up, the intracranial pressure does not go up till much later. So many of these other things, including lactate formation or drop in glucose, etc., occur much before the intracranial pressure starts to go up. So as we're looking at early warning systems, we believe that some of these technologies will take us earlier into and perhaps more sensitively, if that's a word, be able to look at changes in the in what's going on in the brain. You can also look at spreading depression versus glutamate. You all know that glutamate is an excitotoxic neurotransmitter. When it's not good to have a lot of glutamate around, in other words. And when you look at this spreading depression, which is another indicator of brain injury, this is from 2 separate monitors, but you can see that there is spreading depression associated with ischemia, and then extracellular glutamate going up after middle cerebral artery depolarization. So here's sort of a sense of what we have. And when you have a drop in blood flow, the EEG tends to show it early. The spreading depolarization occurs next. Glutamate goes up, glucose goes down, oxygen goes down, lactate goes up, etc. But in a certain sequence, which if this is proven again in the clinical setting, would give us interesting insights as to what leads to what leads to what, and where can we interrupt things, and what can we do to the patient, whether it is cooling or jacking up the blood pressure or taking the bone off to allow the brain to swell. What are the interventions that we can make in order to prevent the bad stuff from happening, or to reverse it, and to reverse it early rather than to wait a day, 2 or 3 when the damage has occurred and there's not really much more to do. So the second generation, we hope, will be able to also study neurotransmitters, will be able to monitor perhaps. And in the third generation, perhaps some protein biomarkers, such as the ones that Dr. Tracy's lab has been working with. And those could also be important in understanding the pathophysiology of this. There are closed-loop continuous samplers. One might be able to do this on a regular basis, not just with CSF, but potentially also do it with things in the blood. We haven't gotten into the blood side yet because that is a whole other undertaking. There's a smart lab on a chip for immunoassays, which Dr. Ahn, who is Chunyan's mentor in Cincinnati, a computer science person, he's been working on developing these microchip, lab-on-a-chip type devices, which will allow us to look at some of these biomarkers. So then to wind up, the smart catheter is capable of continuously monitoring physiological, metabolic, and electrophysiological parameters, while also working as a simple tube to drain excess CSF. With a single device, we were able to demonstrate changes in a bunch of different parameters, as I just showed you. The results further show the importance of multimodal measures of the same tissue to discover cause and effect relationships. I believe we have shown that these biomems technologies are now opening a new era for advanced multimodality monitoring. And the next step, which is a big step for us, is to take this Rube Goldberg type of gizmo and convert it into something that you could take out of a packet and implant into somebody's brain. So that's the next step, and as you can imagine, is a somewhat complex step going through the FDA, et cetera, et cetera. But we're looking forward to making it happen, and hopefully it won't take another 7 years to get to that point. So in conclusion, I'd like to thank our funding sources. This work was supported first by a small grant from Integra, and then a much larger grant from the Department of Defense. And we've got people working here at the Feinstein and our collaborators at the University of Cincinnati. And finally, I'd like to thank Dr. Tracy, Dr. Zura, and others for allowing us to be part of this very exciting symposium and this very exciting institute here at the Feinstein. Thank you.